Good afternoon and welcome. My talk is going to provide some definitions for commonly used terms in this pre-ANDA space, as well as lay the foundation for the talks to come. For a generic product to be substitutable for the reference-listed product, there are three criteria that must be met. The generic must be pharmaceutically equivalent, meaning that it contains the same active ingredients, dosage form, strength, route of administration, and meets the same standards of quality, strength, purity, and identity as the reference-listed product. It must be bioequivalent, which means there should be no significant difference in the rate and extent of absorption of the active ingredient at the site of action. And finally, to be therapeutically equivalent, the approved drug must demonstrate pharmaceutical equivalence, bioequivalence, and then it can be expected to have the same clinical effect and safety profile when administered to patients under the conditions that are specified in the labeling of the reference-listed product. Since we are here to talk about complex drug device products, what is a combination product? 21 CFR 3.2 defines a combination product as any product composed of a drug and device, a biologic and a device, a drug and a biologic, or any combination of the three. The Office of Combination Products has classified 9 categories of combination products. Of these, types 1, 2, 4, and 7 relate to drug-containing combination products and are the most common types we see for genetics. The following slides will show examples of each of these types of classifications. Type 1 combination products are those that are in a convenience kit or co-packaged. These are probably the most widely familiar to the audience, and they include such things as a pre-filled blister pack that contains the vial, the drug, and the syringes and needles required to administer the product. But it also includes such things as medicine droppers, dose counters, measuring spoons, and even rulers and measurements that allow the user to measure out the dose of the drug to be applied. Type 2 combination products are also familiar to this audience. These are the pre-filled drug delivery systems. In this category, the sole purpose of the device is to deliver the drug. And you can see from the illustration some of the commonly used types of drug delivery systems would be transdermal patches, the various inhalers, nasal sprays, pre-filled syringes, and auto-injectors or auto-pens. Type 4 combination products not only include the device and the drug, but the device is coated or impregnated or otherwise combined with the drug. But the device has an additional function in this category in addition to delivering the drug. On the left-hand side, you see the Sinuva nasal implant. In this product, the implant not only delivers a corticosteroid to the site of action, but it also has a mechanical function of maintaining the patency of the nasal passages after surgery. In the middle, you see the estrogen ring, which allows sustained release of contraceptives over time. And another example would be the smart pill, such as seen in the Abilify MySite. And finally, the type 7 combination products, which are less frequent. These are separate products that require cross-labeling. Typically, the NDA is approved at the same time as the device is approved, either through the PMA process or the 510K process. The most common example of these are the light-activated drugs that are not co-packaged with the drug and device, but are labeled specifically for a device. On the left-hand side, you see Cysview, which is used for the identification of bladder cancer with the Karl Storz system. The center is another example from Stryker, which is the spy agent green, an indocyanine green agent, which has to be used with the Sci-Fi system for the detection of lymph nodes during gynecologic surgery. And finally, on the right, you see the 5-ALA or levulinic acid product that's used with the blue U light. Some general principles when evaluating combination products. The performance characteristics take into consideration the performance of the device constituent and its interaction and impact on drug delivery. However, this is not the focus of the comparative analyses, which I will discuss in the following slides. For that, the user interface is the critical piece that we evaluate, and this is the focus and evaluation in a comparative analysis. In January of 2017, the FDA issued the Comparative Analysis and Related Comparative Use Human Factors Studies for Drug-Device Combination Products submitted in an ANDA. And it is this guidance that provides the framework for how the evaluations of a generic product should be done. Please note that this guidance is currently under revision, although has not been completed at this time of the presentation. Some of the key points from the draft guidance include the fact that the generic device does not need to be identical to the RLD. The differences in design of the user interface should be adequately analyzed, scientifically justified, and not otherwise preclude approval under an ANDA. Some design differences in the design of the user interface should be minimized in the early phases of drug development. There are certain labeling differences that would be allowed, but these are considered on a case-by-case basis. The expectation is that the end users will be able to use the generic combination product when it is substituted for the RLD without the interventions of a healthcare provider and/or without additional training prior to use. In addition, we recommend that there be a baseline assessment for any identified differences, and this is done through the comparative analyses. This will determine whether or not additional information and/or data is warranted, and this would typically be in the form of a comparative use human factor study or additional in vivo or in vitro data. This is not intended to demonstrate the safety or effectiveness of the proposed generic combination product. It is assumed that if it's bioequivalent, that it will be safe and effective. So now let's look at some key definitions from the guidance. What is the user interface? Well, that includes all components of a product with which the user interacts. So the labeling and the packaging, the delivery device and its constituent parts, and any associated controls and displays. An external critical design attribute is a feature that directly affects how a user will perform a critical task necessary to use or administer the drug product. And a critical task are those tasks that, if performed incorrectly or not performed at all, would or could cause harm to the patient or the user, where harm is defined to include compromised medical care. What is a comparative analysis? The guidance defines 3 sections to the comparative analysis. The physical comparison, which includes the visual, auditory, tactile examination of the physical features, including size, shape, and any feedback of the reference listed product compared to the same features in the proposed generic drug-device combination product. It's recommended to start with this area because changes in this section may affect how critical tasks are performed and how the labeling will need to be addressed. The comparative task analysis compares step by step each task that is required for the user to perform in order to successfully administer the product. And the labeling comparison includes a side-by-side, line-by-line comparison of the full prescribing information, the instructions for use, and any descriptions of the delivery device constituent parts of the generic drug-device combination product and its RLD. When performing your comparative analyses, in the context of the overall risk profile, there are 3 possible outcomes that can be assigned. One is no difference. The second would be a minor difference, where a difference in the proposed generic interface in comparison to the RLD user interface does not affect an external critical design attribute. An other than minor difference, however, is a difference in the proposed generic user interface that may impact an external critical design attribute that involves administration of the product. So when you've done your comparative analysis, there are 2 outcomes. It can either be complete or incomplete. If the analysis is deemed incomplete, it may involve one or more of the individual sections recommended in the guidance. Some of the common omissions and errors that we see include missing comparative assessments or images, omitted tasks, omitted sections of the IFU, or changes in labeling that are not permissible under the regulations. This table lists some of the more common examples of what we have found in incomplete comparative analyses. The differences may be identified, but they're not categorized as recommended in the guidance. And minor differences and other than minor differences may be identified, but they're not justified. The comparative task analysis, we frequently see that the difference in a physical feature is not linked to how a user will perform a specific task that may be affected by that change in the physical characteristic. Additionally, we're seeing a lot of user-related risk analyses submitted instead of a comparative task analysis, and I will explain the difference in the next slide. For the labeling comparisons, we frequently see such things as omission of preparation to use the drug or cleaning steps that are required. In the upper right-hand corner of the slide, you'll see an example of a URRA. This should be familiar to most of the audience if you've been involved in device development. You identify the task, any potential use errors that could occur while trying to perform that task, and then a characterization of the potential harm that can be caused if it's done incompletely. In addition, you also look at the risk mitigation strategies that you can control to prevent that from happening. In contrast, the comparative analysis shown in the left lower corner is exactly what it says. It is a comparison of each of these features with the RLD and the generic. So how a task is performed in the RLD product should be the same as the comparative task when it's performed in the proposed generic. So what are some of the key takeaways from this talk? A complete comparative analysis includes a physical comparison, a comparative task analysis, and a labeling comparison focused on the instructions for use. During the ANDA review, all of the labeling components are evaluated, but in this pre-ANDA space, only the instructions for use. Pre-ANDA assessment of the comparative analysis can provide feedback as to whether a proposed device may be appropriate for an ANDA submission, and whether there may be any other than minor differences between the user interfaces that might warrant submission of additional data to the FDA to support that the differences don't alter the overall risk profile of the proposed generic when compared to the RLD. Generic product labeling should be the same, although some differences are permissible as described under Regulation 21 CFR 314.94. So our recommendations to the industry would be to make sure you read and understand the draft guidance, the comparative analyses, and relative comparative use human factors studies for a drug-device combination product submitted in ANDA. Throughout the drug-device combination product development, consider your user interface and the critical tasks required to be performed in the RLD product. Evaluate each of the risks associated with the differences between the proposed generic and the RLD user interface. You want to perform iterative comparative analyses to seek to minimize the differences from the RLD. You need to consider whether the user interface in terms of whether they impact an external critical design attribute that involves product administration. If your device design is final, then you need to consider whether any additional data beyond just the comparative analysis would be needed to support or justify any remaining user interface differences. And this, for example, would be whether or not you need to perform a comparative use human factors study. We recommend that you talk early and often with the FDA through controlled correspondences or pre-ANDA meeting requests for complex products. And finally, I'd like to acknowledge the following people for their help in developing this presentation. And with that, thank you for your attention.